Okay, I think we can get started. We still have people joining. Welcome everyone to today's seminars in oncology. Thank you for joining us. And a reminder for anybody needs reminder on Zoom is like, please post your questions in a Q&A in the right-hand side of the screen. And also, the seminar will be recorded and will be available in a website later on today that is listed on the link is on the screen, as you can see. And it's my pleasure to welcome today's speaker, Dana Pear from Memorial Sloan Kettering Cancer Center, where she is the Alan and Sandra Gary Endowed Chair in Computational and System Biology program, and she's also professor of physiology and biophysics at Weill Cornell Graduate School of Medical Sciences. She's also the director of the Single Cell Research Initiative at Sloan Kettering and the director of the Metastasis and Tumor Ecosystem. And Dana is, doesn't really need introduction. She has had an illustrious career. She is originally from Israel, where she got her degree in computer science and also a bachelor degree in mathematics, and then a PhD in computer science as well. And then subsequently, she came to the US and did a postdoc with George Church here at the Harvard Medical School. And then following that, she was a faculty at Columbia University in New York for a while. And then since 2016, she's been at Sloan Kettering. And Dana is a pioneer of devising computational tools for analyzing high-dimensional data, such as the single-cell data, CyTOF single-cell RNA-seq, which nowadays almost everybody is using. And today she will talk about the cellular plasticity in cancer. Thank you, Dana.
Thank you very much for inviting me. That's a lot of fun. Anyway, I'm not in the first slide. There we go. So I like giving a general title so I can talk about whatever my muse is about. Quick disclosure, first and foremost, I'm on the advisory board of In Vitro, but really it has nothing to do with what I'm presenting here. So all you're getting is my opinionated view of things, but with no financial bias. And I'm really enamored with plasticity. Basically, I really think that it's plasticity, cellular plasticity, that's not driven by specific mutations that give its the tumor cell remarkable adaptive abilities, particularly to colonize metastases, to evade the immune system, to develop drug resistance. And a wonderful way to study this plasticity is single cell technologies, particularly single cell genomics. And the reason I fell in love with single cell data, and pretty much ever since I fell in love with it, since CyTOF and later single cell RNA-seq did nothing else, is because single cell data from a tissue really reveals a continuum of states. And if you're interested in cell state transitions and plasticity and how a cell can change, then you really get this continuum directly from a single sample, and this asynchrony between the different cell states really allows you to do a lot. Now, plasticity actually is not a thing that tumors invented. We have to remember that it actually is an innate ability of the cell. It's particularly important during development. Again, I'm going to open with a few oldies but goodies. This is a work I did with Kata Gentanakis in endoderm development. And what you see here is a gut tube during gastrulation. We have the extraembryonic cells in green that are slated to become extraembryonic tissues, such as placenta, umbilical cord. You see them actually being sucked into the gut tube. So actually the gut tube that gives rise to all our internal organs is actually a mosaic of both epiblast embryonic cells, but also extraembryonic cells that sort of found their way in, as you can see in this video. And what happened to these extraembryonic cells as they go into the gut tube, and what's their fate? So we used single cell data, and the first thing we saw that, while many times the top principal component is is pseudo-time. Here, the top component was actually pseudo-space, and the gut tube organized, the single cell data organized along a pseudo-space where each cell was really organized as a cluster. Even the gut tube at this point was very, very uniform, really resembling its promotal organ that's about to bud out from each place along the gut tube, whether it's lung or liver or pancreas. And because we had these cells fate map, we could look at the cells that were of epiblast origin and the cells that were of embryonic origin and see, what did they express? And first of all, if you sort of control for where they are along this pseudo space, where they are along this gut tube, and we did validate this pseudo space, you can see that each, whether it's epiblast or extraembryonic, they all express the master regulators of the organ that is going to bud out of that location. And generally, there was more than 99% correlation between the cells that were of embryonic and extraembryonic origin. And this really points to remarkable plasticity, that even though the cell was intrinsically set out to become extraembryonic, it found itself in the gut tube, looked around, and was able to respond to its neighbors and to external cues and say, everyone's about to become a liver. Let me switch my fates to a liver cell too. Now, cancer really amplifies this plasticity to an enormous degree. And here's another work with Joanne Massaguet, led by Ashley Longyu, who's now at Cornell Weill across the street from me. We really saw that if you look at metastatic lung cancer cells from the clinic, you could really see that one of these axes of progression really recapitulates quite accurately organogenesis of the lung. So you could see all the factors going on in order of lung organogenesis, as well as their downstream transcriptional programs. And it's as if, metastatic cells are trying to develop a lung in the wrong time and in the wrong place, using the very same transcriptional programs that the developing embryo gets. And this is part of the plasticity that the cancer cell has to get access to all sorts of aberrant transcriptional programs. So now I'm going to tell you a few news stories. This is going to be the main story. This is a collaboration with Scott Lowe down the hall from me. We're on the same floor, and it's been really a close collaboration between Cassandra in my lab, a grad student, and Dorena in Scott's lab. Dorena is now on her way to Barcelona to the IRB. And this is truly a close daily collaboration between bench scientists and computer scientists to really try and understand early PDAC initiation. So a lot of people, when they study cancer, there's been a lot of focus, certainly genomically, on the mutation and taking a mutation-centric view to tumor progression. And indeed, the tumor does gain driver mutations with time that makes it, first arrive at malignancy and then increase its malignant abilities. But what happens in this really early pre-malignant phase, where it doesn't seem like there are more driver mutations being added on? A lot's going on, but it doesn't seem to be genetically driven. Of course, and this is one of the reasons I love visiting Dana-Farber, is that I have a lot of like-minded people who think cell lines aren't enough, and you really need to look at these cancer cells in vivo, because there's just so much interaction going on with their microenvironment. And you need to be able to query, the epigenetic changes of these cells, because, something is changing about them, and it's not mutationally. Now, most studies of PDAC, and there's been quite a few published, really profile the endpoints, but it doesn't tell you a lot about what happens early on. So we're going to leverage genetically engineered mouse models to really start and study the earliest steps in tumor genesis, and particularly the interplay between injury and mutation. So again, we're leveraging Scott's wonderful mice, and we want to compare regeneration, or what happens in an injury in a normal wild type mouse, an injury in a mutant mouse. In this mutant mouse, we had a situation where all acinar pancreas cells were KRAS mutant, so oncogenic KRAS, only the acinar cells, but all of them. So we could induce oncogenic KRAS in the acinar cells. These cells were also labeled so we could actually enrich for them. They're a relatively rare population in the pancreas, and we really wanted to do a deep dive into them and look at what happens before injury, immediately after injury. So we're talking about a day or two after injury, and 3 weeks later, 5 weeks later, when pannins arise, and the injury is resolved, but not normally in the mutant mouse, and as well as what happens in later fully malignant tumors and metastasis, which also have p53 loss. So these are our time points, and please remember these color codes, because these colors are going to appear all throughout. And we start with a t-SNE that takes a global view of the entire thing, and we see lots of different cell types. And of course, we're going to delve in and try and understand what's going on. But I just want to give you a slight feel for the data. And this is some of the best single cell data I've ever seen. Dorena's hands are experimental magic. So when we zoom into these cells, first of all, it's important to say that these are very rare cell types. Normal data sets don't include these because they don't enrich so deeply for the epithelial. The other thing to note is every cell is colored by its condition, and there was no harmonization, there was no data integration. This is the normal processing of single cell RNA-seq, and all the replicates overlapped perfectly, and even replicates across time points, as you can see. Really, when you have the same cell type with no data fudging, normalization, or anything, just really aligned, showing the very clean cell handling that Dorena was able to do. Now, the PDACs, the more malignant, they were each different. So we had 3 different cancers, and each of them took a completely different region of the map. Now, because we have such an overlap in the normal cells, we know that this is not an artifact. And in fact, we do have a small shared state where each of the 3 tumors share this one phenotype. So again, stressing that this is not artifact. But we really wanted to understand the epithelial cells, and we wanted to understand the oncogenic epithelial cells. So first of all, I said that this data seems to have progression. So diffusion components is one of my favorite approaches to look at the progression. So I looked at the first diffusion component. It really shows you the major progression, the major transition in the data. And it was really nice and encouraging to see that on the left right here, we have these completely normal wild type regeneration. On the right, we have the nasty malignant cells. And then we have an axis of progression where these early stages are somewhere in between, showing us that this major axis of progression is capturing some biology. And before delving and going crazy studying a mouse model, you want to sort of really get your bearing and get some common sense comparison to convince yourself that this somehow matches human. So we actually collected a couple PDAC from the clinic. And here again, these were enriched for epithelial cells, so we could really capture the epithelial cells quite well. And we could see that even in PDAC, this sort of progression matched. So now we're only going to zoom in on the KRAS cells, and we're actually going to use a force-directed layout visual, which is a much better layout to see progressions. The layout is much more faithful to the diffusion components. But again, I want to stress, never over-interpret a 2D visual, even when it is more effective. And this really, you can sort of see this diffusion component, from left to the right, really matching a more pre-malignant group of cells and a malignant group of cells. So let's try and see what's going on. First of all, we see that even very early on, KRAS mutant cells and KRAS a day or two after injury are very heterogeneous in their own right, and they're a bit of a mess. We don't know what other states are initiating, where cells are going, what directions are happening here. This is particularly hard to infer directionality in a system like cancer, where all our assumptions of normal development and normal behavior have to be thrown out the window. To our aid, we're going to take something called CellRank, which I recently developed together with Fabian Theis and a graduate student that came to visit in my lab for a year, Marius Lang. And this is really marrying the best of both worlds of sort of these graph manifold-based things that really keep the data within the manifold, and all these more classical pseudotime algorithms that really take a global view into account. And RNA velocity, which was invented, just down the street from you, which really gives you some clue of directionality, but it's noisy and local. You combine that with lots of good math. And I want to stress that in the cancer setting, and this is a mistake I see a lot of people making, you can't really use velocity in cancer, because velocity really captures timescales of 8 to 12 hours, not more than that. If you're trying to see, well, what happens between time point 1 and time point 2, 3 weeks later, you really can't do that. So here we're only going to focus on where we have day 0, day 1, and day 2 in these very early time points where the velocity is still valid. So we ran our velocity algorithm, and one of the real big advantages of velocity is, again, it uses this math to automatically infer, without any prior knowledge, what are the initiating points? What are the sources from which there seems to be cells transitioning out of, but nothing going in? So these are sort of the initial points based on sort of the mathematical model. And we see that, there's not 1 point, but there's actually 4 points. And this actually reconciles the field, because there's been a lot of lineage tracing results, and this person says it's NES plus, and this person says it's PTFA1, and everyone sort of argues what the initial state is. And the answer is it's actually all of them, and we really could organize all these different lineage tracing works with single markers at a time. So now we can map our origin to our data, so we could sort of orient ourselves with these 2 origins, and we could see what's happening. And under injury-induced activation, cells seem to go more PDAC-like, and we can also measure these distances in the nearest neighbor graph and in high dimensional, again, never to overinterpret what we see in 2D. And there seems to be an acinar to a PDAC-like bridge that seems to go almost all the way to the shared PDAC state. And there's actually a lot of similarity between the cells in this bridge and the shared PDAC cell state, but not quite. There seems to be breaks. These breaks are called P53. But the big surprise to us came when we looked at the pannons, when we looked 3 weeks later and 5 weeks later, when the inflammation clears, and it seems like the signatures actually revert back and become farther away from a cancer-like state. And again, this is all validated with metrics in the high dimensional space as well. And we wanted to understand what's going on here in this sort of dynamics of the system. And we know that a lot of this is really due to chromatin remodeling. So we wanted to bring chromatin into the picture. And at first we took chromatin, the bulk chromatin that was published here by Direna's paper recently published in Nature. And we added some bulk ATAC-seq of the pannons as well, so we could see what's going on with them and how they compare to the early injury in the adenocarcinoma. And one of our big surprises was, again, very similar to our force-directed layout. It seems like the injury is some state that resembles both pannon and PDAC, and it's in the middle. It almost seems like a divergence of routes to a more benign pannon and to an adenocarcinoma rather than a more linear model. And if you actually look at the peaks that are open, you see that there are clear pannon peaks, and you see that there are clear PDAC peaks. But the injury state in blue up here seems to have both pannon open chromatin and advanced metastatic PDAC open chromatin. And again, these are cells 1-2 days after injury. This is very, very early on. And we already see chromatin states that resemble, and peaks that are open that we find open in advanced and metastatic PDAC. So we wanted to look at this further and see what's going on in early stages, what's going on in the blue. So we decided to use one of my favorite tools, classification. And we built a classifier which says, okay, let's look at the expression of a single cell and tell me what stage it came from. Is it a pannon, or is it a primary PDAC, or what is it? And we saw that this, it's a very easy classification problem. And we saw that, we had almost perfect recovery in our test sample. But now we said, okay, instead of applying it to real late stages, let's apply it to that early 2-day injury stage and see how these early stages, both the uninjured and early injury stages, classify. And what we see here is actually that all of them, classify either as priming towards a penin-like state or to a PDAC-like state. And we see that many of the early KRAS cells that aren't injured are more primed to a benign state. And we see that after injury, there's an enrichment for cells that are more primed towards adenocarcinoma, but not exclusively. And the more interesting cells are these intermediate cells right here, which seem to be primed in both directions. They don't seem to be clearly primed to one or the other, but seem to have more plasticity in both fates possible. Again, zooming in on these cells that are an intermediate in their probability, they really are the most plastic states, and they align exactly at our origins here. So our origins are most plastic, and they sort of don't quite know whether they have sort of propensity in priming to be both panin and PDAC. So we wanted to sort of dig into that a bit more. So, we decided now it really justifies to collect single cell ATAC-seq, and single cell ATAC-seq recapitulated, unsurprisingly, all the major populations as before. And we saw the same type of priming that we saw in the RNA in the ATAC-seq as well. Now, rather than sort of just do standard analysis, we decided let's try and use this classification idea again. But instead of trying to classify cells based on, RNA, let's classify them based on ATAC-seq. So now we look at all the different subpopulations and all the different clusters. And actually, we had a much higher resolution of clusters than what I'm showing you here. And let's build a classifier for what cluster you're in, for what cell state you're in. And this is, transcription to transcription, RNA-seq to RNA-seq, and that works really well and really accurately. But now let's run the classifier that we ran and developed on RNA on the ATAC data, basically looking at the openness of a gene as its a proxy to its activity. So now, basically, we quantify the amount of open chromatin at each gene and use that instead of its expression, and use the same classifier we built with RNA. Now here we have open chromatin for the red cell, so it will classify as a red cell. The green cell will classify as a green cell, and that works beautifully for these very strongly primed cells. But we also see cells that have open chromatin for multiple states. These are cells that are more plastic. We don't know if they're blue or if they're red. And indeed, in the classification, it comes back with uncertainty. It comes back with, well, I have some probability to be blue, and I have some probability to be red. And these cells will also have a much higher entropy in this probability. And using this, we can actually build a classic confusion matrix for all the clusters that we found in our single cell ATAC-seq and all the clusters we found in our single cell RNA-seq, and see, okay, how do these map? And we see that there's a whole bunch of transcriptional states which are just not explained by chromatin dynamics, that none of the epigenetic clusters seem to score very highly for them. And it seems that these might be driven by something environmental that's not quite captured in the epigenetic data. Even more interested in are these plastic states, these clusters of ATAC-seq that seem to have propensity to go towards and to classify towards more than one transcriptional state, that they have open chromatin that really resembles quite a few different transcriptional states. And that's really what we wanted to look at. Now, one of the advantages here is that this gives us a quantitative measure. If we look at the entropy of this classification and look at our certainty of how many cell types we can get and how certain we are about these cell types, really, this is a probability distribution we can compute entropy on, that can give us a proxy measure to plasticity. And now we can say, okay, what are the populations that are most plastic? And it's these progenitor cells, these origin cells that we've been looking at this whole time that also seem to be, primed both direction, as well as some tuft cells. But now we have for each individual cell a measure of its plasticity, a measure of its uncertainty and its sort of fate. And so we can see what else is going on in these cells because we have a full, transcriptome for them and a full epigenome. And what we see is that they are most strongly enriched. The top enrichment by GSCA, like by a big gap, not cherry picked, is cytokine receptor interaction. So plasticity is really associated with propensity to communicate with the microenvironment. Quick look, we also collected the CD45 plus immune cells, and we see that even though these cells are 100% wild type, the KRAS mutation was only in the pancreatic acinar cells, we see huge shifts in both the myeloid and in the T cell lineages, and cell states accumulate and deplete, and really a massive remodeling of the immune cells even a day or two after injury. And we see a lot of differences in the receptors and ligands that are really expressed in this data set. So we decided to look at the receptors and the ligands and the communication modules a lot more systematically. And what we saw, and this is again looking back at the KRAS mutant epithelial cells, is that there was just a lot of open chromatin at all these cells, and that these cells actually broke down into modules. And we wanted to break it up into modules, but as you can see, there's a lot of clusters that we can see going on the diagonal, but there's a lot of activity off diagonal. So we didn't want a one-to-one correspondence. We wanted to capture also the activity and the correlations that we see, all this red off diagonal redness. So we built a sort of an algorithm to build these communication modules where a gene can belong to more than one module. And these communication modules really define the sender and receiver potential of these cells. And this really organized our cells quite nicely into different ranges. I mean, there was the progenitor module, the gastric module, the tuft module, and each of them really was defined by a whole set of co-varying receptor and ligand expression. Not every cell expressed all of these. There's huge heterogeneity within each of these modules, but even though there was huge heterogeneity in each of these modules, there's still quite significant co-expression of these genes, as you can see from the matrix. So this is in the RAS mutant cells. These are all these early cells that are, no injury, 1-2 days after injury, or the pannins. Let's look at the wild type cells under regeneration, so both normal and in injury. And we don't see as much communication. I mean, this is like a colorful rainbow, and these are pretty dark, pretty black. There's not much going on. The wild type regeneration is very, very quiet in terms of its receptor ligand interactions, except for this region right here. So we wanted to see, okay, what are these regions right here that seem to have this sort of gastric module program open? Looking at what these cells are, these cells actually have a very, very, very high activity for the KRAS signature itself. So even though KRAS here is wild type, these cells, due to the injury, these are all the cells in the injury state, in the regeneration state, they are transiently expressing KRAS, and it really further supports the fact that all these communication modules, all these sort of openness of the chromatin of both receptors and ligands is really driven by KRAS. In the wild type, much less so, and much more transiently. So now that we know, okay, there's so many possible communication, let's break it down. And here we're looking at the potential crosstalk between these receptors and ligands between all these different modules. We did something somewhat similar to cell phone DB, but while cell phone DB is looking for potential communication between cells, we're actually looking for potential communication between these gene-based communication modules that we found. But the most important thing we did here, from my perspective, is we said, we're only going to count a link if it's unique to the oncogenic KRAS setting. If this link absolutely doesn't exist in the wild type, and this is a new type of communication that's only enabled in the mutant KRAS setting. And now you could see, what this sort of, I call it a fuzzball. I hate these sort of fuzzball graphs, but what you can learn from this fuzzball graph is that there's just so many new communications that are only possible in the KRAS mutant between epithelial cells and their environment. The gray cells are immune modules, and that there's a bunch of modules that are sort of master epithelial modules. Every one of these balls represents the number of such communications that exists. And you can see that these few modules have just a ton of communications, new communication with the immune system that just doesn't exist in a wild type setting. The interesting thing about these master epithelial modules is that these are the guys that are open in the advanced PDAC. They're absolutely most open and most active in the shared PDAC-like state, which I told you exists for every single one of the three tumors that we found. And even more importantly, we went back to our human data, and I'm not going to show you the exact data because it's pretty ugly, but very statistically significant. These modules are also observed in the human PDAC sample. So there's, again, these differences, these communication modules, these new avenues of communication are not only in these early injury states, but they also exist in advanced mouse PDAC and in clinical human PDAC. So now, again, we want to understand these modules, and I personally can't understand a thing from these fuzzballs. So I like looking at it as a matrix of who can talk with who and how many connections do they have with them, giving each module a sending score, how many signals can it send, and a receiving score. And again, the gastric module is a winner in both the number of signals it could send to its microenvironment and the number of signals it can receive. Of course, you have to go down to the genes. Everything's about the genes, and you can see that there is indeed an enormous number of cytokines and receptors at play here in both the gastric module, the more Pannin-like module, as well as the progenitor module, these initiating states that we saw at the tips, that sort of were the initial states by our cell rank algorithm. And many of these same genes, these are the genes that were plasticity associated. These are the genes that were highly correlated to whether a cell was plastic or not. So we assumed that these were the most important genes because, our plasticity score said, these are the most plastic cells. And so the cytokines that are expressed in the most plastic cells might have a role. And we noticed that, again, here in this point, we're cherry picking, and I think it's legitimate to cherry pick based on your biological knowledge. We noticed IL-33, which was a big player in Dorena's recent paper, which really showed that IL-33 alone induces an injury-like metaplasia in the KRAS setting. So if you throw IL-33 with no actual injury on the KRAS mutant, you get morphology that looks very much like what you'd get if you'd actually injured the cell. And so we decided to really zoom in and focus and understand how is IL-33 communicating with its environment, with what other cells and through what. And so we decided to build a sort of IL-33 centric model where we're focusing on all the direct interactions of IL-33, as well as the further downstream indirect modules of IL-33. And we see that IL-33 is sort of expressed in sort of gastric Pannin-like module, and that its receptor is actually in the Tregs and the ILCs in the microenvironment. These Tregs and ILCs not only are able to get the IL-33 receptor, they're expressing IL-4, which is able to signal back both into the epithelial compartment as well as the macrophage compartment. And IL-4 receptors we see on both the gastric and the progenitor modules. The progenitor module is able to receive a lot of signal explaining why it's so plastic. And again, these two modules are very active in the shared PDAC cluster. I just want to really stress that. And again, this is very KRAS mutant specific. If we're looking at the regeneration context, if we're looking at wild type and injured wild type, there's no IL-33 receptor. There's no IL-33. There's no IL-4 receptors. There's nothing to send or receive the signal, which actually initiates a feedback loop with the immune system that rapidly and dramatically remodels both the epithelial tissue and the immune system together. So again, we want to study IL-33, and the fun thing with working with Scott Lowe's lab is that they have these speedy mice. And so if we want to perturb something, it's still a lot of work. You still have to wait longer than a computational scientist likes waiting because we're an impatient bunch. But now we could build a special mouse that can sort of spatially and temporally control when and where we're knocking down IL-33. And we're going to knock IL-33 down only in these sort of acinar epithelial cells, nowhere else. And we can knock it down, at will when we decide to knock it down, and see what happens when we knock down IL-33 in these epithelial pancreatic acinar cells. When we looked early on, and here I'm using Milo, which is my favorite algorithm to look at how populations change in single cell data. I think a lot of the approaches out there are very much flawed. And Milo is just a great tool developed by the Marioni lab that we now use almost exclusively to study changes in populations after perturbation or between treatments or cohorts. And really wanted to see, okay, how does the population structure change between the regular experience, the regular KRAS situation, and KRAS plus knockdown of IL-33. And we see that early on, it's actually the immune system that's showing a lot of dramatic and a lot of significant changes. The epithelial compartment, not so much. So we actually perturb the epithelial compartment and create a dramatic remodeling of the immune compartment 1 and 2 days after injury. But when we look towards injury resolution, when we look at what happens 3 weeks later, we see that there's not that big of a change to the immune system, but actually there's a very dramatic and substantial change in the epithelium compartment. So zooming in, we wanted to see, okay, how does the epithelial change 3 weeks after injury? What are the populations that are changing, and how do they map back to the populations that we've been talking about all along? So now we can map each of these populations to all our different communication modules that we've been talking about and that we've mapped earlier. We have the progenitor module, something downstream of the progenitor module, the gastric model, and we can see where are the biggest changes. And the biggest changes are indeed in these 2 meta hubs of the gastric and progenitor master epithelial modules. And again, we map each cell. I mean, these modules are really defined by genes, but we can map these gene programs to cell, asking what is the gene program that the cell is most strongly expressing. So the cells that are most strongly expressing this gastric program right here, these cells are being heavily depleted in the perturbation, in the IL-33 perturbation. And we can now really quantify that and attach a significance and say, okay, we had this module of direct and indirect and non-interacting populations with IL-33. The direct interactions changed the most. Their population frequencies changed the most dramatically and most frequently. There is an impact on the indirect, not as much as the direct, but quite significant. And there's no impact on the non-interacting modules, really showing that the modules that we predicted to respond based on the cells that we predicted to respond based on our communication modules are really the ones that are responding. And what we can see is that we have these progenitor cells, these get depleted, they move forward into this sort of downstream progenitor state that usually continues into the gastric pannon-like state, but in the IL-33 perturbations, they seem to get stuck. So to really summarize the story, what we really see is that the KRAS mutation induces cellular plasticity through mechanisms. First of all, KRAS is able to completely remodel and change the epigenetic landscape dramatically. And part of this remodeling involves the opening of a lot of chromatin regions around both receptors and ligands. And under inflammation, this completely remodels the crosstalk with the immune system, causing a dramatic remodeling to both the immune and epithelial compartments. Now I'm going to take a tiny little detour. And in this tiny detour, I'm going to tell you about an algorithm that we're about to release into the world. This is really led by Sitara and Manu in my lab in a collaboration with my better half, Itzik. And the idea is, and I'm going to take this, little foray just to show you, because we're going to use meta cells in the next paper. We actually used them a little bit in this paper, but I swept that under the rug, the first one. And the idea of meta cells, and I think they're most needed in ATAC-seq, but they also help RNA-seq, is that at the single cell level, single cell ATAC-seq is way too sparse to do anything. And you can't even distinguish noise from signal and what peak's a real peak. And so what people do is they cluster it. But once you cluster it, it becomes way too coarse, way too blunt to really capture these more subtle transitions where you could see here, the peak is opening slowly, but you don't see that when you sort of average out these too big of a population. So the idea of metacells, and we can run them, and we have our C-cell algorithm for this, is to really capture the sort of most tight state. These are cell states that are really represent the same biological state, that really we think that the difference between the expression or the open chromatin that we see is driven by sampling and technical reasons and not biological noise. And this really allows us to enumerate the biological states in our system in a sort of a more comprehensive way, unaffected by cell density. And now that we can aggregate all the cells in a state, we also get much more robust both gene expression and attack profiles for this state that we've sort of worked out. Now, again, a quick benchmarking just to show why we're so excited by the C-cells algorithm. We put a lot of effort for the C-cell algorithm to really uniformly capture all the states. And when I say uniform, it's unaffected by density. One of the things that I strive for in my algorithms is to develop algorithms that are able to capture the rare cell types. A lot of algorithms just go for the density. Almost all of machine learning is to try and do maximum likelihood of the data. So you're going to sort of overlearn dense regions and completely neglect the rare and infrequent cell populations. But all the good biology, whether it's stem cells, whether it's the metastasis initiating cells, whether it's transient state between one state to another, all the biological goodness is in the rare cells. And that's what we focus C-cells on, to really be comprehensive and get all cell states, including the rare ones. Now, once you have C-cells, I just want to give you a little bit of flavor of how powerful this approach is. We can really now correlate between open chromatin and between transcriptional state. And you see these beautiful, and this is bone marrow data to benchmark where we know the answer, and you could see the high correlation between a single peak and the expression of that gene. And of course, it's being, the TAL1 is being expressed in erythroid and RF8 in dendritic cells. And just to show you how ugly it is in single cell data, I mean, it's garbage. There's no correlation. And I can do, this is not cherry picked. I can do pages upon pages. And there's just so much that we could do with these metacells. And they also really help in aggregation of RNA data, which I'll show you here. Now, this is fresh out. So we put out these two preprints on bioRxiv this weekend. So anyone who wants to can read this work. This has been a wonderful and still ongoing collaboration with Charles Sawyers, with a big team of fellows. On my lab, everything's being led computationally by Joe Chan. Samir Zaidi actually took this as an opportunity to learn computational analysis and how to do so rigorously. So he certainly did a lot of heavy lifting on the computational analysis as well in Charles's lab. And of course, these two completely gifted experimentalists, particularly Wouter, who I'm going to talk more about his work. Now, we're going to look at lineage plasticity in prostate cancer in organoid models. That's all Wouter. And in mouse, GEMS, and in clinical samples, and that's Samir and Jimmy. These are the two papers, sort of organoid and mouse human. First of all, looking at some clinical samples of advanced drug-resistant metastatic prostate cancer, we found an even bigger heterogeneity than we expected. And we expected a lot of heterogeneity. I mean, you always sort of hear that, wow, single cell discovers heterogeneity. This one even surprised us coming into it, expecting heterogeneity. And particularly within each patient, we saw heterogeneity in AR expression. That's the target for the drug, even though these are all supposed to be drug resistant, as well as heterogeneity in expression of even different transcription factors that drive many neuroendocrine cell types, an A or an N subtype. So we decided, okay, we have to understand this plasticity. Looking at the clinical samples is too complex. Let's try and look at simpler models. And I love the organoid module because the organoid model really pares it down to its most simple form, and it's highly perturbable. And Wouter built this great prostate organoid model where you can study things. And, inducing P53 and RB loss, these are the two. So again, when you have P53 and RB loss, then these cells are able to avoid androgen therapy and completely switch from and revert to a different, more neuroendocrine lineage and become drug resistant. So it's really the tumors that have P53 and RB loss are the ones that are able to sort of develop drug resistance through lineage transformation. But of course, not all of them do. Something else is going on. What else is going on? So we decided to induce plasticity by deleting these two genes or inducing the deletion of these two genes in these prostate models. And we see that after about 4 weeks, we get this hyperplasia where these organoids become dark and filled in. And after 8 weeks, we even get these ugly mesenchymal slithering-like phenotypes, as you could see here. So we have this sort of pretty normal phenotype, this sort of ugly filled-in phenotype, and the EMT slithering one. Now, we saw that actually, and we were surprised, both the basal and the luminal cells, if we sort of sorted them separately, are able to give rise to this plasticity. And this double deletion of these two genes are just really potent. Whether you're luminal or basal, about 90% of the cells develop these sort of plastic organoids following the co-loss of these two genes. So we decided, okay, now let's see what's going on transcriptionally. Let's dig in and look at the transcriptome of these different states. And again, we also quantified, and we saw that at 2 weeks, the organoids are still completely cystic and nice and pretty looking. And they almost all of them, 90% of them, reverted to hyperplastic at 4 weeks. So we see the quantification. Slithering, took a while. Now, when we look transcriptionally, we saw that the wild type is very nicely organized, and at the transcriptional level, really organizes into 5 distinct clusters. 3 of them very clearly basal. 2 of them very clearly luminal. And so you have these 5 very distinct clusters. You have a clear distinction between basal and luminal transcriptional states. And even at 2 weeks, while the organoid is morphologically cystic and normal looking, you have a complete mess up, and you sort of lose a lack of identity, a lack of separation between luminal and basal identity, which only continues to get worse. And what you're seeing here is every single dot here represents metal cells. So this is sort of a summary statistics showing all the states that we see in the system, just to sort of really pare down and get a better view on what are all the different cell states that we observe in the system. So now in all these images and a lot of our analysis, each dot is going to be a metacell computed by our C-cell algorithm that really computes what are all the comprehensive state of cells in each one of these conditions. And then we can sort of aggregate these profiles as I showed you earlier. Now, we wanted to classify each cell. And here we used a slightly different classification algorithm, which we find really to work really well. This is a graph-based classification algorithm based on the neighbor graph, showing how easy it is to get absorbed into each one of these states. And we basically decided to look at the most frequent states, 2 basal states and 1 luminal state. And we could really quantify or classify things. Some cells were clearly luminal, some states were clearly basal, and that's a quantification we were able to get. And again, at week 2, everything was clearly basal or luminal. We lost and sort of got mixed phenotypes. I couldn't decide whether they were basal or luminal. They had both basal and luminal features, both basal and luminal markers, which only got worse and further mixed as we progressed in the time series. And we could actually quantify this uncertainty, again, this entropy. As a proxy to plasticity, we could quantify the entropy or uncertainty of classification. Another really great way to sort of look at this mixing is to look at a graph-based approach. And again, the organoid here really splits into 2 regions. The neighbor graph of the metacells either is luminal or basal with very few ways or routes to transition between each of them. And every cell is sort of really either clearly luminal and blue or clearly basal and red. 2 weeks in, remember the organoid is still normal looking, but transcriptionally, things aren't normal. There's a lot of yellow mixed states and seemingly many more paths of potential transdifferentiation between these two lineages. And by week 4, everything is a total and complete mess. Almost all cells are in between luminal and basal. Very few have clear basal and luminal identity. And you seem to have like almost no barrier in any which way path to go between luminal and basal. Again, we can use sort of diffusion map and really see how many paths are there between basal and luminal in these sort of neighbor graphs, and quantify that as yet another measure of plasticity, which corresponds and completely matches our previously measure of plasticity. Of course, we can't directly measure plasticity without lineage tracing, without seeing what are the future progeny of an individual cell. But we've developed for this work 4 different metrics of plasticity that sort of have some common sense features, and they all agree. We asked, what are the causes of this plasticity? Here is really wonderful to have a full transcriptome so we could see what pathways correlate with cells that are highly plastic, that score high on the plasticity score. And we see LIF signaling and JAK-STAT signaling as the top 2 most enriched pathways that also get based on what's most plastic at an early stage at week 2, because we saw that plasticity is induced at week 2. So we wanted things that go up at week 2. We also see, for example, FGFR going up very strongly. And here just to show you how JAK-STAT signaling is really highly correlated with this basal luminal mixing entropy score. And it's highly correlated with all these other scores as well. But this is an organoid. We're talking about JAK-STAT and interferon and all this good stuff, but there's no immune system here. It's all autocrine. So what's going on? Well, actually, the fact that it's all autocrine makes life a little bit easier in a way, because you don't have to figure out which cell is talking with cell. You could look at the ligand and the receptor in the same cell. So you basically said, okay, if this ligand and receptor are really active in this autocrine loop, we expect to see very high levels of JAK-STAT and interferon signaling in cells that have high level of the receptor and the ligand in that same cell. So we could look at the ligand, the receptor, and the downstream signaling in the same cell and actually develop a score. And again, LIF and IL-6, for JAK-STAT, as well as a lot of FGFs, are really, really high. So we look at all these most significant ones, but again, we have that added filter of knowing when this is happening, because this score is globally across all time points, across week 4, across week 8. And we know that this initiates at week 2. So we only looked at the things that really change between wild type and mutant P53RB1, and already changed significantly at week 2. And that really gave us LIF upstream of JAK-STAT and FGF. Those are the 2 top predictions. And we saw that the inhibition of each of these and most effectively the combined one really reverted the organoid to normal. So again, we quantified whether it's cystic and nice looking or hyperplastic and ugly looking. And we saw that whether we inhibited either one pharmacologically, it had an effect, and we reverted to the cystic phenotype. And both of them had this enormously synergistic effect here, with 80% of the organoids reverting. And we saw the same thing also with both pharmacological and genetic CRISPR inhibition. And that really makes me feel good anytime computational predictions are validated. Looking at the mouse to see, what's going on here, we saw that that the neuroendocrine, the full neuroendocrine state, only occurs in vivo. And we see multiple neuroendocrine states with a lot of similarity to small cell. Again, the nice thing about this sort of transitional thing is we could sort of work out an inferred, assumed pseudotime. We see that the source seems to be these transformed luminal L2-like cells. And we could even look and see what's happening along the pseudotime and see these transitional TFs that really resemble many programs from gastrointestinal development. So the same story as in lung, where they revert back to lung organogenesis. here we see some gastric organogenesis in these prostate cancers that are sort of doing lineage switching and lineage transformation. Again, this JAK-STAT signaling that we saw in the organoid, really high in that intermediate transitional population that's expressing all the developmental programs as well, both in the mouse, and of course, it's much noisier to look at this from clinical samples, but we see this sort of same increase in JAK-STAT signaling as the cells are transitioning from AR positive to AR negative. And again, just to sort of really show how conserved this is, we see really the same programs in prostate patients and in small cell lung cancer patients, really arising at these sort of same neuroendocrine states driven by these different transcription factors. We thought, okay, if we reverted to normal physiology, if the cells are now, less plastic and more clearly basal or luminal, can we revert the drug resistance? And that's what made us really happy, that, following pharmacological inhibition of both JAK and FGFR, that blocks the plasticity and waiting a few weeks for the organoid to revert to normal, we also managed to reverse the drug resistance and create androgen therapy response. And so this is a real therapeutic opportunity. We've managed to really find a drug that stops plasticity in its tracks, reverts drug resistance. This is, of course, in an organoid model. We're very far from the clinic. You need AR signaling, some AR signaling to still be there. If AR is completely gone, it won't work. But early enough, even though most of the AR signaling is gone, you can really revert this state. So really, here's another story of consequences of plasticity where non-genetic adaptation allows the cell to really develop drug resistance through lineage transformation, again, getting access to these development and organogenesis programs. And it's really driven by inflammatory signaling, particularly JAK-STAT. And it's driven in a cell intrinsic way. It doesn't even need the immune system to drive this JAK-STAT signaling. And most fun, we can revert this plasticity with both inhibition of JAK and FGFR. So I talked about some of the causes of plasticity, mutations, aberrant epigenetics and inflammation, and the horrible consequences of plasticity, which I think really is one of the hallmarks of cancer that gives the tumor its most powerful adaptive capabilities. So, acknowledgements in my lab, this was really spearheaded by Cassandra and Joe, two enormously talented. Cassandra's a student, Joe is a postdoc. I talked about some of Manu's work. He's now at Fred Hutch. Close collaboration with Dorena, who's amazing at Barcelona. Wouter, who's on his way to EPFL, and a whole bunch of amazing colleagues. And thank you very much for your attention.
Okay. Thanks, Tana. That was really amazing. I love the pictures and the stories. It's really nice. So
I knew you would like it. I know you.
yeah, I have many questions, but let's start with the audience first. One question by Wubing Zhang is, how did you estimate the chromatin state for each gene? Do you consider attack peaks in each gene? And the question is basically, why is the injury-induced states are not well explained by the single-cell ATAC-seq data?
Okay, so those are two packed questions. Basically, we looked at the chromatin region surrounding each gene and saw how much of it is open in ATAC-seq. There's a lot of details of how we process the data to get a cleaner read of which peaks we believe are real peaks and relevant peaks, because this data is very messy. So there's a lot of very ugly technical details that, it would take me about 10 minutes to go through, but we're writing them up and we're hoping to post that in a bioarchive in the coming month. But basically, you do careful cleanup of the messy data, you aggregate into metacells, and then, look at how much is open in the nearby region of each gene, and you use that as a measure of the activity of that gene. So why is there no explanation? So unlike the module, the communication modules, which I allowed more than one gene to belong to one module and sort of allowed a lot of flexibility. Here we built a clustering, and of course clusters, this was fairly coarse, so we went to the most coarse states. And then classification gives you a probability. So if we were to do a completely different analysis, of course, there is open chromatin that could create the transcriptional states that we saw. You can't create transcriptional states without open chromatin, but it's so not dominant in the data that, you know, neither the clustering algorithm nor the classification algorithm picked it up as significant, because there seems to be so many other factors at play. The chromatin signal for that state was very weak, but not non-existent, obviously. If we would have went the other way around, what cells possibly have these genes open, of course, we would find cells, and we do.
Mm-hmm. A follow-up question is, what factors really regulate plasticity? Are those the cytokine regulation in like what you described in a pancreatic cancer, or is it JAK-STAT? Or I actually think that two are linked. So, but anyway,
Yeah.
let's hear your answer.
So, you know, biology is complex, and, you know, always to try and pinpoint it and say there's one factor. I mean, biology is so many things that are so interconnected. And, you know, JAK-STAT in interferon signaling is directly related to inflammation. And inflammation basically means, okay, we're just, from my interpretation, inflammation means help, we're injured, help, we're in stress. Let's, you know, open a lot of communication receptors. Let's open a lot of possibilities. Let's open a lot of plasticity to fix it. Now, JAK-STAT is one mechanism that activates the interferon and sort of activates this plasticity. Now, of course, JAK-STAT and interferon signaling is a very complex thing that involves many players and many sort of parallel and interconnected and cross-talking pathways. IL-33, for example, seems to be like a very strong initiator. It's the first step, and then it just like opens up a lot of stuff. So basically it triggers a cascade of events that eventually open a lot of these things, including, I mean, of course, we see a lot of JAK-STAT signaling in the plastic pancreas cells as well. But here we tried to tease it apart really down to more specific receptors and ligands. And IL-33 seems to be this massive difference between KRAS mutant and not KRAS mutant that's very dramatic and seems to have a lot of communication loops and cascades downstream of it.
Mm-hmm. Okay, and I mean, we have, maybe I have a question also, because I was intrigued by two things. Was very intriguing. One is that the injury response was different in a wild type and a RAS mutant, so that you got very differences. So that is very exciting in a way, and kind of asked me, like, in humans, germline variation, particularly, you know, variation in cancer predisposing genes, not necessarily mutation, but even variations. How would that influence tissue injury response, and how could that be linked to cancer risk?
So, you know, just, you know, when you break it down and when you actually look at genes, there is a shared gene program between, you know, wild type regeneration and mutant regeneration. So it's not that there's no similarity between them and there's no shared gene program. And there are actually a small fraction of KRAS mutant cells that actually behave normal. Because one of the things that we see about all these aberrant communication modules and programs, they are actually very heterogeneous in terms of what are open in what cell where in the mutant scenario. But generally, we see that the wild type and the KRAS mutant are far more different than they are similar. It's not one or two small differences. It's massive difference. And I think this is the reason why, you know, all these nasty cancers. I mean, pancreatitis is there's a lot. You know, cancer emerges from pancreatitis, possibly because there are some KRAS cells in the pancreas somewhere that's initiating this. Also, all sorts of you know, Crohn's disease and IBD is a predisposing factor for colon cancer. So clearly, inflammation or extended inflammation. First of all, it's a mutagen in itself, and there's a lot of cells there. And if you get some mutant KRAS somewhere in some cell, then you might be in serious trouble. But and also we see that, you know, I mean, obviously at the end, if you have KRAS mutant, that you get a pattern, you never get full resolution. So even, you know, growth phenotypically and histologically, there's differences. But yeah, I'm you know, this work has really gotten me down a path of wanting to look at more and more normal regeneration in mouse and humans, and more on the impact of different mutations on this regenerative process. And certainly a direction that I want to go into pretty strongly. Or started
Yeah,
to.
I mean, I have lots of ideas on that. Maybe I have to tell you, but there's maybe one last question from the audience, from Ken Gray. There's a question about the C cells algorithm. If processes like differentiation or regeneration involve phenotypic continuum, can discrete meta cells really capture all possible states? And if not, can you quantify information loss, or is it not meaningful loss as long as you produce testable hypotheses? So that's a long question.
So there's actually a pretty simple answer to it. So imagine if you had sampled less cells. Imagine if we were still in, I mean, one of the things that MetaCells tries to do is it tries to say, you know what, you know, in single cell RNA-seq, we're only capturing 5% of the transcriptome. In ATAC-seq, we're capturing, you know, a fraction of a percent of the open peaks. So what C cells and the MetaCells approach more generally, because there's a couple different algorithms for this, says is, you know, there's so much technical noise here. If I see these two cells, they're more likely to be different because of this technical sampling than a real biological difference. So the fact that I have two of these cells, they're likely in the same cell. It's not real biological variability. And we do see paths along, you know, you could do pseudotime, and we do pseudotime on MetaCells. And actually, when we run pseudotime algorithms on MetaCells, we get much more robust and much cleaner continuums. So you can do continuums, and you get all these, if you've sampled enough cells, you get these continuums. And on the other hand, once upon a time when we worked with Fluidigm, or people who still work with SmartSeq and these plates, because you want to go deep and you want to get, you know, long reads and all these other things, you get less cells, you get less transitional states. But if you sample it well, you can get transitional metacells, and we do get transitional metacells. And when we use the sort of C cell approach to study hematopoiesis, a clearly continuous process, we get much cleaner results than when we used the trajectories inferred from single cells. So we actually, in the paper, which you'll be able to read in about a month, we did extensive benchmarking of pseudotime on single cells and pseudotime on C cells. And, you know, pseudotime on C cells is better, and you do get continuums.
Okay, thank you so much. We ran out of time. We could discuss this for a long time. I have other questions as well, but thank you for this very, very stimulating talk and the beautiful pictures and explanations. So thank you again. Thanks.
It's been a pleasure, and thanks for listening.
Bye. Bye-bye.
Bye.